Good morning, everyone, and thank you for the kind invitation to present my research work today. My name is Arcangela Iuso. I'm a scientist at the Helmut Center and Technical University in Munich, where I work on MPAN and MPAN-related disorders. In the field of MPAN, I focus mainly on MPAN, where I try to understand the molecular mechanisms leading to the disease, testing therapeutic approaches, implementing existing models, and identifying biomarkers using a multi-omics approach. But I focus also in MPAN. I recently characterized a model of the disease, a mouse model, and currently I'm exploring the molecular basis of mitochondrial dysfunction and testing therapeutic strategies. But I do work also on NBIA-related disorders, namely PPCS deficiency. This is cardiac disorders, but shares with CoPAN and PCAN defective biosynthesis of the coenzyme A. So my goal is to generate and characterize models of this disease, understand the link, the functional link with PCAN and CoPAN, and testing eventually therapeutic strategies. But today I'm going to focus on MPAN, on a specific project. And the aims of this specific project were two. One was to identify what happens in MPAN cells that leads to the disease. And the other aim was to correct the problem using chemical compounds. Before entering into the details of the project, I would like just to give an overview of what we know about MPAN and C19-12. So MPAN arises from mutation in one copy of the gene C19-12 or both copies of the gene. In the first case, specific mutation cause a dominant form of MPAN. But when both copies of the gene are mutated, then we have the recessive form of MPAN. But from a clinical point of view, there's no difference between the dominant and recessive form. Concerning the function C19-12, we know very little. And so we hypothesize that C19-12 is relevant for lipid metabolism because we have noticed that the expression of this gene is particularly high in tissue which are rich in lipids, such as the adipose tissues, as you can see here in the orange, dark orange, and in the clear orange bars, and in the brain, as you can see in the yellow bars. And we also know that C19-12 is localized in different organelles of the cells, mainly in mitochondria. As you can see from these pictures, we have labeled C19-12 protein in green. We have labeled in red mitochondria. And if you superimpose a few pictures, you will see that the two signals almost co-localize perfectly. As I said, the localization of C19-12 is mainly mitochondria, but the protein can also be localized in the endoplasmic reticulum, which is another organelle which is tightly connected to the endoplasmic reticulum, to the mitochondria, and it's involved in the synthesis of lipids and proteins. So, if C19-12 is localized mainly in mitochondria, we can speculate that it's relevant for mitochondrial function. So the absence of C19-12 protein or mutation in the gene would eventually lead to mitochondrial dysfunction. And usually we can follow mitochondrial dysfunction through certain readouts. For instance, we have calcium dysregulation or increased production of ROS. ROS are molecules extremely reactive that damage proteins, lipids, and structure within the cells. And usually, if the ROS production increase, there is a decrease of the antioxidant defense, meaning molecules that counteract the action of the ROS. But also, if there is mitochondrial dysfunction, then this organelle is less capable to produce energy, and usually the molecule which contains more energy is ATP, and also some processes are reduced in the mitochondria, in particular the mitochondrial respiration. So the process through which mitochondria consume oxygen and produce ATP. Sometimes we might have also morphology change in mitochondria when there's dysfunction. So mitochondria gets more fragmented or extremely elongated. And another parameter that is affected by mitochondrial dysfunction is the autophagy. So autophagy is a basic process, fundamental process, through which cells get rid of dysfunctional protein and organelles. So what we did in our project was to collect a large number of fibroblasts from MPAN patients with autosomal recessive and dominant inheritance, carrying different mutations, and investigate the parameters that I mentioned before, and compare these values to the values obtained in control cells. As a control cells, we mainly used fibroblasts from parents of MPAN patients, but also unrelated and healthy controls. So let's start. Let's check what happens in the calcium. So here, each one of these bars represents a different cell line, and the number corresponds to the ID of each fibroblast. As you can see, the values of calcium in the patient cell lines is not so different from those of control cell line. You can distinguish the patient and control cell lines by the color. So the patient are indicated in orange and the controls in green. So to get the better pictures, what we did is to pull all the controls data and pull all the cases, the patient's data, and to compare them. And again, as you can see, there's a slight tiny difference, but this increase is not significant. To calculate, so obviously we use some tools that allows us to determine whether a change is relevant or not. So in this case, applying this statistical tool, we saw that this change is not significant. And so it means that this difference we observe is the effect of a biological variation. What we did next was to stress the cells in a way that the calcium became more relevant for cell metabolism. So why we follow, I did not tell you why we follow calcium. So calcium is, this ion is very important for the communication between the organelles within the cell. And the mitochondria are the main reservoir of calcium. And if we stress cells, we expect to see an increase of calcium release. So we wanted to see whether in this situation of stress for the cells, there was an increased calcium release. Again, we checked each individual cell line. As you can see here, there is just one cell line that stems from the other. And this patient cell line has a mutation which was not represented in the other patient cell lines. So if we do now this pooling of the values, the control and the patient values, you can see here that there is a star. So usually we use this annotation to indicate that the difference we see is significant. But what happens if we remove this cell line that we believe it behaves differently from all the others? If we do not include this cell line in the analysis, then control and patient have the same values. So this significance disappears, meaning that this calcium dysregulation is very likely not present in all patient cell lines, but it's something specific for this specific cell line or specific of this mutation. So we went on checking the production of reactive oxygen species. And again, we couldn't see significant differences between controls and patient. So similarly to what we did for the calcium, we did for the ROS. So we stimulated the cells. We induced a stimulus that induced all cells, controls and patient cells, to produce more ROS. And then we compared if under this condition, there is a difference between patient and controls. We noticed that there is like a tendency in the patient to have higher ROS level than controls. But again, our statistical tools told us that this difference was not significant. So to be sure that this result was true, we also checked the antioxidant defense. As I told you before, if there is an increase of ROS, then you would see a decrease of these molecules that counteract ROS. But we measured the antioxidant defense, and we couldn't find any significant decrease in the patient cells. So we conclude that there is also no ROS production in a consistent way. No consistent production of ROS in patient cell lines. Hence, we measured ATP. Again, there's a trend towards a decrease of ATP in the patient, but the difference is not significant. So we used also the mitochondrial respiration. We checked the mitochondrial respiration. And to our surprise, we found that actually the patient respired better than the control cells. So this tells us that there is no mitochondrial dysfunction, and let's say confirms the previous data, according which there is no significant decrease of ATP. We then checked also the morphology of mitochondria, and we used a microscopic technique called electron microscopy. I'm showing here only a few pictures, but we took many more pictures. And the structure that you see here in green are the mitochondria. So to measure the morphology changes in the mitochondria, we had to draw the perimeter of each mitochondria and extract parameters such as mitochondrial length and eccentricity. So the mitochondrial length is basically, if you consider mitochondria as a tube, the length, the measured length of the structure. And the eccentricity tells us if the mitochondria is like a tube or is more rounded, like a circle. We measure both parameters, but again, comparing control green, with the patient in orange, we couldn't find any significant difference in both parameters. So we concluded there's no significant change in the morphology. And so finally, we investigated the autophagy process. The autophagy process is a process conserved and important for the cells, because this is one of the main mechanisms through which cells get rid of dysfunctional proteins, dysfunctional organelles. And in general, in order to degrade this material, this damaged material gets engulfed in a phagophore, so in structure surrounded by membrane. And during this process, there is a protein which is free in the cytosol, cytosolic protein called LC3-1, that gets activated and then becomes LC3-2 or LC3-B. And this protein binds to the autophagosomes and to the complexes protein and organelles to be degraded. So this is a kind of signaling for the lysosomes, which are other cellular organelles, to fuse with the autophagosome, release enzymes that would cut protein and organelles, and then get rid of this dysfunctional material. And this structure that we obtain after the fusion of the autophagosome with the lysosome take the name of autolysosome. Now, this process takes a certain time normally, but if you want to study this process in vitro, there's a way to fasten the process or to induce it. So we can use autophagy inducers like taurine or rapamycin, but we can also block the autophagy process with baphyllomycin. So the use of inducers or inhibitors or the combined use of both help us understanding where the problem is in the autophagy process, whether at the beginning or at the end in the clearing phase. So this is exactly what we did with MPAN fibroblast. So we try to understand the autophagy flux. So we took controlling patient cells and we used inducers of autophagy, inhibitor of the autophagy, and a combination of both. And without entering into many details, we used some markers of autophagy progression. And we did a quantification of those markers in the different conditions with the treatment. And this analysis told us that in MPAN, the defect is at the beginning of the autophagy process in the activation of LC3 or already in the phagophore formation. So what we did next was to test, to use a different approach to confirm the data that we had already found on autophagy. And we used a microscopy approach. So as you can see here, we have a patient line and also a control cell line and also a control cell line expressing more synaptinol 12. So this is the picture of LC3B when cells are growing under standard condition. So as you can see, you can barely see any green signal. Therefore, we triggered the autophagy process. We damaged mitochondria using CCCP, and we checked how autophagy was progressing, and we followed this LC3B. You can see that in the patient appear some green dots. So this is our LC3 big. But the number of dots that we see in this picture are reduced compared to the number that we see in the control cells. And it's even more reduced compared to cells overexpressing C19-OF12. We did quantification of this signal. And as you can see, there are some stars appearing, meaning that the difference that we calculate this time are significant. So, namely, so in the patient cell lines, so there is autophagy. This is a fundamental process. Cells cannot live without autophagy. But the process is not so efficient as in control healthy cells. So, considering this, we decided to search the literature for molecules stimulating autophagy. So, we selected 15 different compounds and tested them on the patient cell lines. Only 5 of them rescued autophagy. And here in this picture, this microscopy picture, you can clearly see that the patient cell lines have only few green spots, but when we add each one of these 5 compounds, carbamazepine, paroxetine, LY294002, ABT737, and oridomine, the number of these green dots increases and reaches the levels that we have in the control cells. And now, always by checking the literature, if we look at the stage in which this molecule act, they all act at the beginning. So, coming back to the aims of the project. So we wanted to identify the problem in MPAN cells. And we discovered that one of the problems in these cells is that the initial stages of the autophagy are impaired. We wanted to correct the problem with chemical compounds, and we identified 5 compounds that restore autophagy in MPAN fibroblasts, and 2 of these compounds, paroxetine and carbamazepine, are already approved by the FDA, meaning that they could be more easily used for compassionate use or used in future clinical trial. So despite it's not clear whether the defect in autophagy is a primary defect in MPAN or is a consequence of something else, for instance, impaired lipid metabolism, we have now a readout, and we can try to modulate this readout, and hoping that acting on autophagy, we can maybe delay the progression of the disease or eventually stop it. So I really hope I explained clearly enough my project. If you want to know more details, you can read the publication that was that appeared at the beginning of the year on pharmaceutics. And before concluding the presentation, I would like to thank you, all the patients and their families, and all the collaborators that have this project bring some results. And last but not least important, the support, the financial support of the patient organization, NBIA Swiss, NBIA Poland, and Hoffnungsbaum, and the funding from Aerobioimaging for electron microscopy experiment. With this, I conclude my presentation, and I'm happy to take your questions. Thank you.